Hi, everybody. It's Gina. I'm just going to make this quick video. I don't know how quick it's going to be. I'm not super quick at anything, but I want to make this video on protein synthesis. And I think I envision it more as a reinforcement because you've already gone through the handout and answered those questions and went through the Google slides and used those to help you answer the questions in the handout. And if you read this chapter in the new version of the textbook, then I think you'd be ready for this. So hopefully you've done all of those things. You can watch this first and then you can watch it again, whatever you want to do. I'm just not going to teach everything here. Okay. So I'm going to go and do a little drawing of protein synthesis. That's the goal here, to do a drawing. So I'm going to get myself out of the way. I hear my puppy, but my puppy is being obnoxious right now. Okay, so if we're going to make a protein, we have to start with a strand of DNA, because proteins, all the proteins that we make are coded on our DNA, on our genes, right? So we have all these genes that make our chromosomes, and they code for the proteins that we need. So let's make believe you just ate a donut and you have a lot of sugar in your system, your blood sugar is high. So you need to make insulin, let's say, for example. If you're an insulin maker, right? Some folks don't make insulin. Diabetics generally don't have the ability to make insulin, or the shape is wrong. So we'll make believe we're making insulin today. So we need to start with a gene, right? So a gene is made of DNA, and DNA has two strands. It's a double-stranded molecule. Okay, so I'm going to draw a double strand of DNA. I'm going to at least attempt to here. Hopefully that's showing up on our screen. And here's the two strands. So we're going to call this DNA, right? And where we are right now is in the nucleus of a cell. Okay, so we'll get that going. So this is the nucleus of a cell. And we need some code because we can't really do this without code. So we have to have some code. So let's put some in there. I'm gonna make this up. Okay, I'm just gonna make all this up. So we're gonna have a double-stranded piece of DNA. So we're going to have E, A, C. Let's just make this easy for us to do. G, 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 A, T, E, G, G, E, T, 5, 6, 7, 8, 9, 10, 11. And we'll do a G at the G there. Okay. I'm gonna let's see. Oh, that works. Oh, I lost a little bit of that I didn't want to, but that's okay. Put our line back in there. I'm gonna make the last one. Let's see. What am I gonna? Okay, so I just fixed a couple of these. I just ended this with ATC, right? The very end is ATC. And then, so that's one side of the DNA strand. And this is from last chapter, right? This chapter is really about making RNA, but this chapter is about making proteins, RNA and proteins. Last chapter was about double-stranded DNA. So this part, what I'm about to do, you should be able to do, right? This should not be difficult. So if this is the one strand of DNA, the other strand, you should code that in there, okay? So if this says T, this is going to say A. So we're making DNA. We haven't even started making RNA yet. So this is DNA. That's a G. G, 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 C, 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 A, G, G, C, C, A, T, A, G. So there's our double-stranded DNA. And we'll make believe that this is the gene for insulin. It's a little more complicated than that, but we don't really need to get too deep into it. But there's actually code before the start of the gene, and there's code after the start of the gene, after the end of the gene. These are called untranslated regions. There's promoters and things like that. There's all sorts of interesting things that start at the beginning and end at the end, but we don't really need to worry too much about those, okay? So what's going to happen now, this is the first step in protein synthesis. What I'm about to do is step one. First step in protein synthesis is going to be what's called transcription, right? So we're going to do transcription right now. And I'm going to write transcription. And this happens in the nucleus. And it happens where it is the time when we make a copy of DNA onto a molecule of messenger RNA, right? DNA is not allowed out of the nucleus. So clearly something has to happen because protein synthesis happens in the cytoplasm on a ribosome. So DNA is not allowed out of the nucleus. So we're going to make a copy of the gene so that can go out of the cytoplasm and head to the nucleus, head to the ribosome and make a protein. Okay. So we're going to make a molecule of messenger RNA. If you went through all the stuff, you know that there are 3 kinds of RNA: messenger RNA, transfer RNA, and ribosomal RNA, which is just the ribosome. So we need to make the message, right? DNA has the message. We need to make a copy of the message. So we're going to do that right now. So we're going to make a copy of this message. And I'm just going to say, just for the sake of making this easier, that I'm just going to tell you, I'll always tell you that we're going to copy this strand here, this bottom strand. We're going to call that the coding strand. And I'll always tell you which one to copy. And we're going to make a copy of this onto a single strand of RNA. RNA is single-stranded, not double-stranded like DNA. So I'm just going to use the code here and use the DNA to RNA base pair rule. So the DNA to RNA base pair rule is if there's a C in the DNA, you get a G, right? C to G's always stay the same. The only difference here is that RNA doesn't have a T. There's no thymine in RNA. There's uracil. So you can't lay down any T. You can only lay down U. So if you wanted to put a T, stop yourself in the RNA and put in a U. Okay, so let's just practice that a little bit. So T gets an A, right? Because there's A's in RNA. So we're making messenger RNA. So we got plenty of A's, we got plenty of C's, we got plenty of G's, but we don't have any T. So we have to put U instead. Okay, so now there's an A in the DNA. That would mean that we have to put down a U. Okay, C gets a G. All these C's get G's. All these G's get C's. Those ones are easy. T gets an A. G, G, C, C. T gets an A. Oh, there's a U. There's T gets an A and C gets a G. Okay, so we have just made our messenger RNA strand. Boom. I'm going to ask you to do that on the last exam, 100%, without a doubt, going to ask you to do what I just did. The next step that actually happens here is called, and I'm not going to draw this, this is in the handout, is called pre-messenger RNA processing. So this strand that we just made is sort of a premature messenger RNA strand. It's not in its perfect form. If this were to be sent out into the cytoplasm, it would get degraded way too fast. It doesn't have any tags on it to tell it where to go. So one of the things that has to happen to this is it has to get modified to become a mature messenger RNA. One of the first thing that happens is this thing called a 5' G cap has to put on. It's just a guanine cap that's on the 5' end. So this is the 5' end of the RNA strand, and this is the 3' end of the RNA strand. We're not going to really worry too much about that. But a little cap gets put on the front, a big long tail gets put on the end, which is kind of funny. This big long tail of A's, it's called a poly A tail, gets put on the end. And that also makes it so that the molecule knows where to go. And then the other thing that happens here, and this is in the handout, is that sections of the messenger RNA that don't code, we'll do one. I'll do one here. We'll make a mature. So let's make believe this section right here might have to change. Maybe if I could figure out how to do the colors on this, I'll do. There we go. I'll make them pink. So let's make believe this little section here is something called an interon, right? We'll make believe that that's an interon, and that everything else here, what color? Let's go. I don't know if this is a good color, but yeah, that's not a good color. Everything else here is called an exon. Okay, so we'll make this red stuff an exon, just for the sake of it. Okay, so the interons are parts of the strand of messenger RNA that we don't want, right? There's nothing wrong with it. It's just that we don't want. And the red part are the exons. Okay, we want those. So this way I always remember it. Exons get spliced together and exit the nucleus. Interons get spliced out and stay in the nucleus, right? So that's how I always remember. Interons stay in the nucleus. exons are the important ones, and they go out of the nucleus. So now what's going to happen is we're going to get rid of that interon and recreate our messenger RNA. So now I just have to write it again. AUG GGG AGGC C AGGCC AUAG. Okay, beautiful. 3 prime. There is our mature messenger RNA. Okay, so this is the messenger RNA. This really is called the pre-RNA. Okay, so we did all that, and now we can exit the nucleus. We can exit the nucleus and search for our friend called the ribosome. All right, so ooh, that worked. Okay, let's see. Get rid of you. All right, so let's draw ourselves a nucleus here. I mean, a ribosome here. I'll make the ribosome green just first because I like green. Now, this is not drawn to scale. Ribosomes are itsy bitsy. They're super, super tiny. They are much smaller than I have drawn here, but I'm just trying to make this easy for us. So this is the rRNA, right? What we know as a ribosome. We learned about those. Ribosome. We learned about those in chapter 4, right? So we know that ribosomes are the site of protein synthesis. They're the place where all of this is going to happen. Okay, so this is the location for where our messenger RNA strand is going to go. So whoop, we're going to come out. We're going to come out of here. We're going to come out of here, and we're going to find this ribosome. So I'm going to snake this out, and I'm going to draw it on top of here. Okay. And I'm going to rewrite it again. A, U, G, 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 A, G, A, U, A, G. Okay, so that's our messenger RNA. Our messenger RNA is on the ribosome. What I would do, if I were you, this is what I do because my brain can't handle all those letters. It's just too difficult for my brain. I would, because the ribosome reads that code in groups of 3 called codons. I would immediately just break this into groups of 3. Just use a line and break this into groups of 3 just to make it manageable for your mind, right? Just to make it manageable for your head. And so these groups of 3 are called codons. I'm not going to write codon across all of these, but I'm just going to do it once. So this is called a codon. And so somebody along the line, I can't remember, Mendelssohn, I think his name is, discovered that the way in which messenger RNA is read in groups of 3, and those groups of 3 codon, that group of 3 nucleotide codon relates to a specific amino acid, right? So, which is kind of amazing that they figured that out. Okay, so we know that these are read in groups of 3 called codons. And so there's 2 things that have to happen now, and it's hard to teach them both at the exact same time. So I'm going to teach one thing and I'm going to teach the other thing. First, I'm going to teach how you use that codon dictionary to look up which amino acid is brought. And then I have to teach the idea of how to use a transfer RNA. So both of those things are really important. It's just that it's hard to do both at the exact same time. I'm going to try, but I'm going to focus on one and then the other. Okay, so we can see here that the first 3-letter codon, there's 3 nucleotides, makes 1 codon. And you can see the letters are AUG. That's called the start codon. And for most organisms, the gene starts at AUG. The coding part of the gene starts at AUG. So that's really helpful, right? If somebody gives you a big long strand of DNA and then asks you to transcribe it and translate it, you have to start at AUG. You can't start before the AUG. You have to start at the AUG. So we're gonna start there. I didn't give you anything before the AUG, and I probably won't. Wouldn't make it that much more difficult. So AUG is our start codon. We're going to see that there's going to be some termination codons also, or stop codons. Okay, so we'll start with the transfer RNA. So transfer RNA is just another type of RNA. We've already had messenger RNA. We have ribosomal RNA, or the rRNA. Now we have transfer RNA. So I'm just going to make believe that I know how to draw transfer RNA. we'll make transfer RNA blue. Okay, so I like to draw transfer RNA like a T, because it kind of does look like a T in its actual conformation or form, its native form. And so it has two sides to it. It has the top side where the amino acid gets attached to. Oh, that's kind of funny. Moodle realized I raised my hand. Not Moodle, Zoom. Transfer RNA has the top side where the amino acid gets attached, and the bottom side where there's the anticodon. Okay, so let's draw that in there. So the top is where the amino acid goes, and the bottom is what we call the anticodon. Right? So here we go. This is something I would ask you to do. If the messenger RNA codon is AUG, what would be the anticodon on the tRNA? Let's label this tRNA. And it's just the complement, right? So if A is on the messenger RNA, this first one will be a U. If U is there, this one will be an A. And if a G is here, this one will be a C. So the anticodon for this transfer RNA is UAC. That's the anticodon. Okay. And then that will tell the codon, and the anticodon will tells the transfer RNA which amino acid to bring, right? So that's sort of a protection for us, because this is really important. If this goes wrong and the wrong amino acids get placed in the wrong sequence or the wrong order, and then the protein gets made, because they're in the wrong sequence and in the wrong order, the protein doesn't work, because one of the most important things you probably should have hopefully learned in biology is that shape is important. The shape of the molecule, especially proteins, is important. If the enzyme's active site is not the right shape, then the molecule doesn't work. So when this sequence goes awry, right, and something goes wrong, when there's a mutation in the DNA that leads to the sequence being wrong here, then I'm going to bring the wrong amino acids. So tRNAs and messenger RNA make sure that it's like a check to make sure that we're bringing the right amino acid. So if we look at AUG on the messenger RNA, that is our codon. That is what we read to determine which amino acid is going to be brought. So I copied in this little sucker that you're gonna have to be able to use. Let's see, can I make it bigger? Okay, so AUG is our codon. That's the one we got to figure out. So here is the first letter is A. So that means we're going to look at any one of these. All of those start with A. Then I go to the second letter. Well, that's a G. So that means I'm coming down and I'm only looking at these, right? And then the third letter is, oh, I screwed that up. Sorry about that. I apologize. Let's go backwards. Let's try that again. Let's see if we can get rid of this. Oh, it doesn't like it when I use my finger. I don't want to have to remake this whole video. So we're going to just use a different letter. I don't know why it won't let me take that color off there. There we go. We're going to start over. We're just going to start over. Oh, okay. Here we go. Ready? A-U-G. First letter is A. So that makes us look at all of these here. All of these start with A. A, 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 A. And then the next letter is U. So we're going to go into this box because all of these are AU. You can see AU, AU, AU. And then we have G is our third letter. And so that gives us this. There we go. We found it. AUG, which is what we call the start codon. And we can see that the amino acid is MET. I'm going to go back here. Let's squeeze this back down a little bit. There we go. We're going to go back here. AUG is our codon. UAC is the anticodon. They match. And that means that this tRNA is going to have MET. So we figured that out from the dictionary. So I'm going to try to draw this in here. Have patience with me. All right, so this would be UAC. That's the anticodon. And then here's our cute little tRNA. And attached to it is MET. That is our first, that's our first amino acid. We made an amino acid. Yay. Okay, the next one is going to be the next tRNA that comes in. I'll do this one and then you guys can do the next two by yourself. The next one that comes in is going to be the anticodon, the tRNA that comes in is going to be C. Draw this little sucker. All right, so now we got to figure out what the amino acid is. This is a fatal flaw that students do have when they do this. They don't remember which one to read, the codon or the anticodon. The codon. This is called a codon dictionary. This is the codon dictionary, not the anticodon dictionary. You always read the codons. So our codon is GGG. So G's are here. These are all G's. And then our second letter is G. So GGG. There it is, GGG. There's glycine. Okay, GLY. Even if you don't know what the letters mean, no big deal. All right, so GLY. That is our second amino acid. And when the two are next to each other, they bond with what's called a peptide bond. Okay, so I'm going to just pause this for a second, or you pause it, and then see if you can get the next two, and then I'm going to do it. So everybody pause and try to do the next two on your own. There's no point in just going forward without trying, because I know how to do it. You're the one who has to know how to do it. All right, hit pause. Okay, hopefully you did those and you got ARG for the third codon and proline or PRO for the fourth amino acid. And then so the AGG codon brings the ARG amino acid. The CCA codon brings the PRO amino acid. And then you bond them together with those peptide bonds. And then we have this last one. This last one is different, right? This last one, if you look up UAG, if you look up UAG in the codon dictionary, you can, that's bright. If you look up UAG in the codon dictionary, you can see that that will bring what's called a stop codon, right? You can see that stop codon there, UAG. There are three stop codons, UAG, UAA, and UGA are all stop codons. That just means stop, right? There's no such thing as an amino acid named stop. So it's not one of the amino acids. It's just a termination sequence that says, okay, we're done, right? So the last thing that's going to happen here is just that this strand is done, right? It gets broken off. So this is just a stop. This just says, halt, we're done. No more. We don't need to do any more work here. So we're going to stop, right? So stop. And so in this case, our protein that we made is 4 amino acids long. Even though there are 1, 2, 3, 4, 5 codons, the actual protein itself is only 4 amino acids long. There's no such thing as that, but we can't do this forever. And then this just gets released and the ribosome continues to do it over and over again until it doesn't need to do it anymore. So that's the basics of how to make a a protein. I hope this helped you. I hope this worked for you. And if you need any extra help from that, I am more than happy to help. I don't know if having this image is helpful for you. You could just take a screenshot of it. I'm sure you are all smart and able to do stuff like that better than I can. But if I can figure out how to make a PDF of this, I certainly will, and I'll post it online. All right, so let's see here. I am going to stop the share here, and oops, sorry. Maybe I will, maybe I won't. Stop share. There we go. Stop share there. And if you need anything else, just let me know. All right. Have a good one. Bye.